see it?
Yes, ma'am.
Okay, so last class where we stopped? Do you remember in this slide or where? I forgot actually.
This slide, ma'am.
This one?
Yes, ma'am. Yes.
Only this one? Okay. So actually we started the membrane lipid. Is it?
Yes, miss.
Yes, ma'am.
Okay then, so let us start. We are learning about the macromolecule lipid, and we have already completed the storage form of the lipid, which is the triacylglycerol, and another one is the wax. And then the second most important lipid is the membrane lipid, and under the membrane lipid we have learned that there are mainly two types. One is the phospholipid, another one is the sphingolipid. So we will actually look at the structure of the phospholipid and the sphingolipid, and the very first one, which we are actually in general we can say phospholipid, especially if there is the phosphate group is present in the polar head group, then we call it phospholipid. Under the phospholipid, according to this flow chart, you can see under this there are glycerophospholipids and sphingolipid. So first we will look at the glycerophospholipid, which are also known as phosphoglycerides, and actually it will be much more easier if you look at this chart. So these are the general structure of glycerophospholipids, and you can look at this structure where we can see the glycerol, which is the alcohol, 1, 2, 3, the 3 carbons of glycerol, and the 2 carbons hydroxyl group of glycerol, they are linked to 2 fatty acids by ester linkage. Okay, so these are the ester linkage where 2 fatty acids are attached to the glycerols number 1 and 2 carbon, and the number 3 carbon is actually attached to a phosphate group, and further the phosphate group is attached to a head group, which is usually a polar head group. Okay, and when the head group, which we here it is represented by the letter X, the X is replaced with a proton, that is H, then we called it the parent compound of glycerophospholipid, phosphatidic acid. Okay, phosphatidic acid. And now the phosphatidic acid, that means the proton or the hydrogen, it is replaced with other polar head groups. So the polar head groups can be different functional groups or different molecules such as amino acid, then sugar residues or oligosaccharides can be present. So for example, we can say ethanol amine. So ethanol amine is a polar head group. If that is attached to the phosphate, then we call the glycerophospholipid phosphatidyl ethanol amine. Then the polar head group can be choline, and if the choline is present, then we call it phosphatidyl choline. Even amino acid serine can be a polar head group, then we call the glycerophospholipid phosphatidyl serine. Even another glycerol moiety can function as polar head group, and the name of the glycerophospholipid is phosphatidyl glycerol. And there other glycerophospholipids where sugar moieties can be present as the polar head group. So from this table, you can see the name of the glycerophospholipid and name of the polar head group and what is the structure of the polar head group and what is the charge of this glycerophospholipid. Okay, so this chart is actually very important because later when we will learn about the biosynthesis of the lipids, this will help you a lot. Okay, so here actually if you look at the structure, general formula, you just need to remember the glycerol, which is made up of 3 carbons, and then the glycerol's 2 carbons, they are linked to 2 fatty acids, and the third one is linked to a phosphate group, and then the phosphate group is linked to a polar head group. In this structure, at the carbon number 3, the phosphate group comes middle, that means middle position, which is connecting. At one hand it has glycerol, at other hand it has a polar head group, and this bond is known as phosphodiester bond. Is it clear? This bond, the third carbon of the glycerol, linked to the polar head group by phosphodiester bond, whereas the fatty acids, 2 fatty acids are linked to the other 2 carbons of the glycerol by ester linkage. Is it clear?
Yes, ma'am.
So you have to understand the bonds, how the different bonds are involved in linking fatty acids and the polar head group. Okay, because again we will come to know about that during the biosynthesis of lipids. Okay, and the polar head groups, they can be polar molecules which are such as amino acids, then maybe sugar residues as well. So according to the polar head groups, the glycerophospholipids are named. We will look at some of the phospholipids one by one. Okay, these phospholipids, can you see the slide? I move to the next slide. Is it visible?
Yes, ma'am, দেখা যাচ্ছে।
Okay, so glycerophospholipids, sometimes, that means the structure we just learned, sometimes there is another bond, then the ester bond. Here in the structure, if you look at the structure, so the previous one where we have seen the fatty acids, 2 fatty acids are linked to the glycerol by ester linkage, but in this case you can see one of the fatty acids that is linked to the glycerol's number 1 carbon by ether linkage. Okay,
Ma'am, next slide
so you can clearly see the difference. Yes,
Next slide.
ম্যাম প্রিভিয়াস স্লাইড তো শো করছে, নেক্সটটা দেখা যাচ্ছে না।
okay. So you are still on the flow chart, glycerophospholipids.
নো ম্যাম,
ম্যাম এখন চেঞ্জ করতে
ম্যাম এখন দেখা যাচ্ছে।
এখন কি দেখছো?
glycerophospholipids।
না, এটা তো ডিসকাস করলাম, তারপরে এখন যে এই পরেরটাই গেলাম, এটা কি দেখা যাচ্ছে?
হ্যাঁ ম্যাম এখন দেখা যাচ্ছে।
গ্লিসারোফসফোলিপিডস, ইথার লিঙ্ক। আমি যদি এখন এটাকে স্লাইড শো দেই তাহলে কি দেখা যায়? ইজ ইট ভিজিবল?
ম্যাম
ম্যাম এখন কি আপনি চেঞ্জ করছেন?
না, আমি এখন স্লাইড শো দিয়েছি।
ম্যাম এখন দেখা যাচ্ছে।
এখন আমরা আছি হচ্ছে গ্লিসারোফসফোলিপিডস উইথ ইথার লিঙ্কড ফ্যাটি অ্যাসিডস। Is it visible?
Yes, ma'am.
Okay, so let me know whether you can see it or not, because maybe I am discussing something and you are looking at a different slide. Then it will be not clear what is the subject we are talking about. So this one is about glycerophospholipids. That means they have almost same structure we learned, where the two carbons, number 1 and 2 carbons of the glycerol link to 2 fatty acids. But in the previous one, we learned that the fatty acids are linked by ester linkage. But here we are seeing one of the fatty acids that is linked to glycerol, specifically the number 1 carbon of the glycerol, by ether linkage. Okay, ether linkage. So here the difference in between these two type of bond, you can clearly see ether linkage, where at the middle the oxygen, and next one is the carbon of the fatty acid, and another one the hydrogen of the glycerol. Okay, whereas the ester linkage, you can see the carboxylic carbon of the fatty acid, it is also there. Okay, but in ether linkage it is not there. So this type of bond is present in this particular glycerophospholipid, but the head group is linked to the glycerol in the same way as we have seen in the previous slide, through a phosphate group, that means by phosphodiester bond. And here the head group is ethanol amine. And this particular glycerophospholipid is called plasmalogen, and plasmalogen is actually present in animal tissues or unicellular organisms. Okay, and further there is one more feature, the fatty acid, which is linked to the number 1 carbon of the glycerol by ether linkage. The 2 carbons, that means the carbon number 1 and carbon number 2 of the very first fatty acid, they have a double bond. Okay, and this type of plasmalogen, especially they are present in animal tissues. Especially we can talk about the vertebrate heart, even unicellular organisms such as halophilic bacteria, ciliated proteases. Okay, they have this special glycerophospholipid which is having ether linked fatty acid. Okay, they have a special function in case of vertebrate, because this type of lipid, it has a particular function which is called platelet activating factor. So that is actually functioning as a signaling molecule, and these lipid released from the blood cell basophils, and then this molecule actually stimulating aggregation of platelets. Platelets is a blood cell, they will be aggregated. This is one function, as well as they also induce the platelets to release a messenger molecule which is called serotonin. Serotonin, we can also say this is functioning as hormone, which actually cause the vasoconstriction. That means blood vessels contraction happens by the influence of serotonin. Is it clear to you?
Yes, ma'am.
So here we mainly learn a special glycerophospholipid which has ether linked fatty acid, and where are they present and what are the functions of these lipids. Clear?
Yes, ma'am.
Okay, now I will move
Ma'am,
to the
এটার এই
next
স্ট্রাকচারটা
slide.
কি শিখতে হবে?
Let me know whether you can see it or not. হ্যাঁ, স্ট্রাকচারটা তো আসলে খুব একটা কঠিন না। কারণ তুমি যদি আগের স্লাইডে ফিরে যাও where the table is there, this one is almost having the same structure, শুধু there is difference in the number 1 carbon of glycerol bonded to the fatty acid, whereas the rest of the structure is same for the previous glycerophospholipids.
ওকে ম্যাম দেখা যাচ্ছে না
Here and here you just don't need to like write the whole structure of the fatty acid. Just you have to mention the line, the zigzag line. The main focus in the structure is about the linkage and the polar head group. Is it clear?
Yes, ma'am.
Okay. So, what are you seeing now? Which slide are you seeing?
And previous
Ma'am,
slide
ethyl link fatty acid.
previous slide টা
ম্যাম প্রিভিয়াসটাই দেখা যাচ্ছে ওই যে ইথার লিঙ্কড ফ্যাটি অ্যাসিড যেটা
Okay. So I am moving to the next. Please let me know whether you can see it or not. Can you see it?
No, ma'am.
No, ma'am.
Not yet.
No, ma'am.
Maybe there is problem now,
Yes, ma'am. দেখা যাচ্ছে।
Yes, ma'am.
but it is not in slideshow. So
No,
if I
ma'am,
make it
slideshow
slideshow,
হচ্ছে না।
then yes, because if I make it slideshow, then I think there is problem. Okay, I think we will carry out with this without slideshow. Okay, so next group of membrane lipid, they are called galactolipids and sulfolipids. So from the name you can say that they are the glycerol under the glycerol lipids, but here these kind of lipids, they don't have any phosphate group, and also here the polar head groups are sugars or sugar derivatives, and these kind of lipids are mainly present in plant cells. Okay, so under the galactolipids specifically, we can say the sugar residue is galactose, and that sugar galactose is linked to the carbon number 3 of the glycerol by glycosidic bond. In the previous one, where we have seen the polar head group is linked to the glycerol by phosphodiester bond, because there is the presence of a phosphate group, but here there is no phosphate group is present. Instead, the sugar is directly linked to the glycerol by glycosidic bond, because to form a glycosidic bond that we learned where two hydroxyl group can interact. So glycerols, now carbon number 3 has the hydroxyl group, and the sugar that also has hydroxyl group. So these two hydroxyl groups, they interact and linked by glycosidic bond and give rise to the lipid which is called galactolipids. So if you look at the structure, the glycerol is the alcohol, and the number 1 and 2 carbons of the glycerol linked to two fatty acids by ester linkage, and then the number 3 carbon of the glycerol is linked to a sugar. Here particularly it is galactose by glycosidic bond. Okay, and we said that these type of lipids are present in plant cells, but what is the particular location in the cell? They are present in the thylakoid membranes, which are the internal membranes of the chloroplast. Okay, that is the location where these type of lipids are present in plant cells, and that actually make up about 70 to 80% of the plant lipids, membrane lipids of the plant cells. Okay. Okay, now next one is the sulfolipids. So very simple, where the hydroxyl groups of the sugar residues are sulfated, then we call it sulfolipids. So same way, the two fatty acids will be linked to the glycerol by what kind of bond? What is the name of the bond here? The fatty acids linked to the glycerol.
Glycosidic.
What? Fatty acids. Fatty acids linked to. Yes, ester.
Ma'am,
Ester.
linkage.
Yes. So fatty acids will be linked to the glycerol by ester linkage, but the sugar residue that is linked to the glycerol by glycosidic bond. Okay, clear? Okay, now the thing is that here when we look at the backbone where the glycerol is linked to two fatty acids, then we call it 1,2-diacyl glycerol. Okay, 1,2-diacyl glycerol, because two fatty acyl chain is linked to glycerol by ester linkage. And suppose in that case there is no head group is attached to the glycerol's number 3 carbon. So at the number 3 position there is only hydroxyl group, but in number 1 and 2 positions there are two fatty acyl chain attached to the glycerol by ester linkage. Then we call that molecule 1,2-diacyl glycerol. Is it clear?
Yes, please.
Okay. So আবারও বলছি, এই যে স্ট্রাকচার গুলো আমরা বলছি, how are these different parts, they are linked to each other. It's very important, because if once you understand this, that will help you in the biosynthesis process as well. Okay. Okay, তাহলে we understand the galactolipids and sulfolipids. Next lipid, membrane lipid, is the archaeal tetraether lipids. Okay, so this type of membrane lipid is present in archaea. And archaea, they are microorganisms, and archaea is able to live in extreme conditions. So extreme conditions means the temperature is very high, the pH is very low, and the ionic strength is very high. And these archaea, their membrane lipids is made up of long chain branched hydrocarbons. So if you look at the structure, the middle part, actually the yellow shadowed part, which is the diphytanyl groups. Okay, so that is the hydrocarbon, long chain branched hydrocarbons, which is almost made up of 32 carbons. And branch কেন বলছি, এই যে যেটা তোমরা চেনটা দেখতে পাচ্ছ, জিগজ্যাগ লাইন, ওখান থেকে আবার কিছু কিছু স্ট্রেট লাইন আমরা দেখতে পাচ্ছি. So those are the branches. Okay, and also at each end they have glycerol, the pink colored part. Those are the glycerols linked to each end of the hydrocarbon chain. Okay, and these glycerols, they are linked to the hydrocarbon chain by ether bond. Okay, so we can see two hydrocarbon chain, they are linked to the glycerols, two carbons, by ether bond. They are not ester linkage, they are ether bond, and these ether bonds are very strong, and because of these they are able to like live in the extreme conditions, because ether bonds cannot be broken down in extreme conditions, like with high temperature, low pH, that means acidic condition, or high ionic strength. Okay, these bonds are stable. So because of this, it is possible for the archaea to live in extreme conditions. And ester linkage, they are not stable in these extreme conditions. Okay, is it clear why the archaea able to live in extreme conditions? Because their membrane is made up of ether lipids rather than ester lipids. Clear?
Yes, ma'am.
And also these lipids is very like they have like huge they are huge in size. That means they are twice the length of phospholipid and sphingolipid, and they almost span the full width of the surface membrane, a single molecule. Okay, the ether lipid. Okay, now if you look at the structure again, so we can see at two end glycerols are present, which are linked to the hydrocarbon chain by ether bond. And further, if you look at this, at one end the glycerol number 3 carbon, that is actually linked to sugar residues. Okay, sugar residues, whereas at other end that glycerol is actually linked to glycerol moiety by a phosphate group. That means there is another phosphodiester bond is also present, and on the other end where the sugar residues are attached, that is bonded by glycosidic bond. Is it clear? আমরা বুঝতে পেরেছি কি?
Yes, please.
Okay, so the main, we can say the main backbone is actually made up of very long chain branched hydrocarbons, and in the structure it is mentioned that that is diphytanyl group, and then that hydrocarbon at the two ends they are linked to glycerol by ether bond, and those glycerols in one end, at one end, that is linked to sugar moieties, and other end that glycerol is linked to another sugar moiety by a phosphate group, which is actually the phosphodiester bond. And another important feature, these glycerol moieties, they are usually present in R configuration. So they have different configurations than the sugars present in the normal glycerophospholipids. Okay, so these are the features of the ether lipids, and they belong to the membrane of the archaea, and because of these lipids the archaea is able to live in extreme conditions. Is it clear?
Yes, ma'am.
Okay, let us move to the next membrane lipid. Actually now we will focus on another group of membrane lipid, which is called sphingolipids. So this is the fourth large class of membrane lipids, but according to the nature of the membrane lipid, where we have learned they have two properties. That means they have the hydrophilic group as well as hydrophobic group. So sphingolipid also has the same features. Okay, now here the polar head group, that means the polar head group is also present, and the hydrophobic group is also present, but the main difference is that there is no glycerol. Okay, no glycerol, but they have two non-polar tails. That means in case of sphingolipid, that non-polar tails belong to the two fatty acids attached to the glycerol by ester linkage. In this case, we will learn what are the two non-polar tails in sphingolipid, but there is no glycerol. Instead of glycerol, what is the backbone of sphingolipid? Actually, the sphingolipid, it has long chain amino alcohol, which is called sphingosine or 4-spinganin, or its derivatives. So that is the backbone of the sphingolipid, and that sphingosine is attached to a long fatty acid chain. Okay, so fatty acid chain and actually the long chain amino alcohol sphingosine, these two belongs to the two non-polar tails of the sphingolipid. Okay, now the polar head group. The polar head group can be the same polar head groups we have seen in case of glycerophospholipid. So they can be linked to the remaining structure of the sphingolipid by phosphodiester bond and glycosidic bond. That depends on the particular polar head group. এটুকু বুঝতে পেরেছি আমরা?
Yes, miss.
আমরা আসলে আরো ক্লিয়ার হব আমার মনে হয় আমরা যদি এই ফ্লো চার্টে চলে যাই। ওকে, সো তাহলে আমরা যখন গ্লিসারোফসফোলিপিড পড়েছিলাম, ইফ ইউ রিমেম্বার, সো এখানে যে আমরা ব্যাকবোনটা দেখতে পাচ্ছি, যেটাকে আমরা পিঙ্ক কালার শেড দিয়েছি, সেটাকে বলছি আমরা স্পিঙ্গোজাইন। আর গ্লিসারোফসফোলিপিড, ইন গ্লিসারোফসফোলিপিড দ্যাট ওয়াজ glycerol. Okay, so sphingosine is a very long chain molecule. That means it has more carbons than the glycerol. So here the sphingosines number 1, 2 and 3 carbons. Okay, number 1, 2 and 3 carbons, these are analogous to the 3 carbons of glycerol. Okay, now the number 2 carbons of the sphingosine is linked to a fatty acyl chain by amide linkage, because the number 2 carbon has an amino group. That amino group interacts with the carboxylic carbon of fatty acid and give rise to an amide bond. Okay, so here the fatty acid is linked to the sphingosine's number 2 carbon by amide linkage. Okay, now comes the number 3. So number 3 carbons is actually linked to the remaining chain of the sphingosine. So these two chain, they belongs to the non-polar tails of the sphingolipid. Is it clear? Non-polar tail kotha theke ashlo? One is the fatty acid, another one is the remaining chain of the amine alcohol sphingosine. Okay, now the polar head group. The polar head group is attached to the number 1 carbon of the sphingolipid. Sorry, the sphingosine. So here is the number 1 carbon. So here actually number 1 carbon of the sphingosine is analogous to the number 3 carbon of glycerol. So in the same way, this X that can be replaced with other atoms or functional group. So if it is replaced with hydrogen, then we call this ceramide. So actually ceramide is the parent compound of sphingolipid. The same way we learn for glycerophospholipid, phosphatidic acid is the parent compound of glycerophospholipid. And here ceramide is the parent compound of sphingolipid. Okay, now next, in the same way we learned in case of glycerophospholipid, the polar head group will be attached to the structure, and that can happen by phosphodiester bond or by glycosidic bond. So phosphodiester bond will be involved when there is the polar head group, such as amino acid or other head groups will be added. For example, here phosphocholine. If you look at the head group, choline actually, that means choline is attached to a phosphate group, which is then finally linked to the carbon number 1 of the sphingosine and give rise to the sphingolipid called sphingomyelin. Okay, sphingomyelin. Okay, now rather than the choline or an amino acid, other polar head groups, specifically sugars, they can also present in sphingolipid. And if sugars are present, then the sugars will be linked to the number 1 carbon of the sphingosine by glycosidic bond. Okay, in this case we will have glycosidic bond, but in case of phosphocholine or amino acid, they will be linked through a phosphate group. That's why that bond is called phosphodiester bond. Okay, so now it depends on the what sugars are present as the polar head group, the name of the sphingolipid, such as if simple sugars are present, then we call them glycolipids. Okay, glycolipids, or they can also have disaccharide, trisaccharide, or even tetrasaccharide. These polar heads groups can be present, or maybe an oligosaccharide present as the polar head group in sphingolipid. So according to what type of sugar moiety or moieties are present, we named the sphingolipid. So they have different name according to the sugar moieties. So we will see little bit elaborately about each of them. So the very first one is sphingomyelin. So here they have almost same polar head group as we have seen in case of glycerophospholipid, which is phosphocholine or phosphoethanol amine. Okay, so because of the similarity in the polar head group, we can actually place these lipids, membrane lipids, under the glycerophospholipid. Okay, because head group is different and head group is same, and the head groups are also attached to the backbone by a phosphate group. That means the bond is called phosphodiester bond. So regarding that part, they have similarity. So we can also place them under the glycerophospholipid as the phospholipid. Okay, now some feature of this phospholipid is that if the polar head group is phosphatidylcholine and they actually have no net charge, and where we can find them, this kind of sphingolipid, they are present in animal cells. That means animal cells plasma membrane have this kind of sphingolipid. Specifically, they are present in the myelin sheath of the neuron, which is actually neuron has a different structure than the normal cell. They have a long projection, and that is called axon, and that axon is surrounded by a membrane, and that membrane is rich in this sphingolipid myelin, or that layer is called myelin, which is rich in this particular sphingolipid known as sphingomyelin. So that layer is called myelin, and from that myelin, actually the name of the sphingolipid come, which we call sphingomyelin. Is it clear?
Yes, ma'am.
Miss. Yes, ma'am.
Okay, so in case of sphingomyelin, we can say sphingomyelins, they belong to sphingolipid, but because they have same polar head group and same type of bond, we can also place them under the glycerophospholipid, or in general just phospholipid. And then what is the nature of the polar head group, there is no net charge, and where we can find it, we can find it in the plasma membrane of animal cell, specifically in the neurons axon. The axon is covered with a sheath which is called myelin, because the myelin is made up of sphingomyelin. So this is one sphingolipid. The next sphingolipid is glycosphingolipid. So from the name we can say that in this lipid the polar head group is sugar. So that means a simple or single sugar can be present as the polar head group, or maybe more than one sugar can be present. So based on the how many sugars are present, the glycosphingolipids are divided into different types. So there is no phosphate is present. So the polar head group sugar, which is linked to the sphingosine number 1 carbon by glycosidic bond. And the types are, number 1 is called cerebrosides. Okay, cerebrosides. So here a single sugar is present as the polar head group, and that sugar can be galactose, and galactose is usually present in the plasma membrane of neural tissue, or glucose can be present as the polar head group, and glucose is present in the plasma membranes of non-neural tissue. So they are called cerebrosides. Okay, another type of glycosphingolipids which are called globosides. Okay, globosides, they have 2 or more sugars. That means when there are more than one sugar is present, we call them globosides. So that's globosides sugar residues are usually D-glucose, D-galactose, N-acetyl D-galactosamine. But obviously there will be more than one sugar residues present in globosides, but the interaction is glycosidic bond. And these 2 types of glycosphingolipids, they are called neutral glycolipids because they don't show any charge at the neutral pH, which is pH 7. So the previous one, sphingomyelin, and these 2 glycosphingolipids, they are neutral, or they show no charge at pH 7. Clear?
Yes, ma'am.
Okay, so let us move to the next sphingolipid, which are called gangliosides. Gangliosides, it has polar head group, but that head group is complex than the other glycosphingolipids. So mainly the polar head group is made up of oligosaccharides. Okay, and example of such oligosaccharide is the N-acetyl neuraminic acid, acylic acid, actually which we have already learned under the glycolipids, if you remember, and that residue is present at the terminal end of the glycolipids. And this particular oligosaccharide, it has a feature because it has a carboxylic group, and that carboxylic group dissociates proton, and then it shows negative charge at neutral pH. Actually, this feature we have already learned during we studied glycoconjugates, the glycolipids, if you remember. Okay, now the gangliosides, if they have only one sialic acid residue, they are called monogangliosides. If there are 2, that is digangliosides. So in this way, we can have like tri, and then 4 can be present. So we can call it the quaternary gangliosides. So based on the how many sialic acid residues are present, they can be further divided into the different ganglioside. Okay, now sphingolipids. So specifically
the head group. Head group is actually present on the outer surface of the plasma membrane, and these head groups are functioning as the sites of biological recognition. Okay, now specifically the blood cells, we have RBC. On the surface of the RBC, the sphingolipids we have, which are actually the gangliosides. And we already come to know the gangliosides, they have this sugar moiety, which is functioning as the polar head group. And that sugar moiety is actually defining our blood groups. Okay, is it clear to all of you?
Is there a problem? Any other questions I can answer?
I'm sorry. My wife here, my beautiful wife, just gets a little paranoid.
Is it clear?
You wouldn't believe how long it took her to just go out with me in the first place.
Yes, ma'am.
Hello. Okay, so if you look at the diagram, so we already have learned the structure of the sphingolipid. So here you can see the sphingosine, and the fatty acid is attached to the sphingosine's number 2 carbon, and then the number 1 carbon which is attached to the oligosaccharide. Okay, so this one we are defining O antigen, because this O antigen is present on the surface of the RBC, the red blood cell. Okay, now how it differs from one another, you can just see at the structure. When we are calling it A antigen, additional sugar residue is attached to the original O antigen. So you can see a rectangular structure is attached to the O antigen, then we called it A antigen. And whereas in the B antigen, a different sugar residue is attached to the O antigen, and then we called it B antigen. Okay, so the thing is that the sugar moieties which are attached to the ceramide, functioning as the polar head group, they have different structures or sugar moieties, individual sugar moieties, and those sugar moieties is actually defining our blood groups. So that is the one of the major function of this sphingolipid, specifically the ganglioside. Even the gangliosides, they function as recognition sites. That means these sugar moieties are recognized by other molecules. They can be extracellular molecules, or maybe they are involved in the interaction with neighboring cells. Okay, so these sphingolipids, which we are learning them from the perspective of membrane lipids, but we actually have focused them before from the glycoconjugates. Under the glycoconjugates, they belong to the glycolipids, and their major function is in the recognition as well as in determining the blood group of animals. Is it clear?
Yes, please.
আমরা বুঝতে পেরেছি?
Yes, ma'am.
So আমরা যদি এখানে আমাদের আন্ডারস্ট্যান্ডিংটা ক্লিয়ার হয়, তাহলে this will help us in our immunology course. Okay, so this will help us in our immunology course if we understand this. Okay, let us move to the next one, and the next membrane lipid is sterol, and they are also, because membrane lipids are structural lipids, but sterols are actually present in eukaryotic cells. Okay, whereas other lipids from the beginning we have seen they can be present in prokaryotes as well, but these sterols, they are usually present in eukaryotic cells. And the structure of the sterols is different from the membrane lipid we learned so far, because the sterol has circular or ring structure. We call that steroid nucleus, and that nucleus is made up of 4 fused rings. If you look at the structure, 1, 2, 3, 4, A, B, C, D, designated by A, B, C, D. So among the 4, 3 has 6 membered rings and 1 has 5 membered ring. Okay, A, B, C, these 3 has 6 membered rings, and D has 5 membered ring. And the most common example of sterol is cholesterol. And sterol also has the same feature as membrane lipid, which is they are amphipathic. That means they have hydrophobic part as well as hydrophilic part. So if you look at the structure of the sterol here, the hydrophilic part, that means the polar head group, is actually coming from this hydroxyl group attached to the number 3 carbon of the A ring. So that is the hydrophilic part or the polar head group of the sterol. And the remaining part, that is the nucleus, that is the ring structure, and then we can see the hydrocarbon side chain, all those belong to the hydrophobic part of the sterol. And that side chain, we called it alkyl side chain, and that can be made up of up to carbon number 17. Sorry, carbon number here it is 27. And this side chain is linked to the carbon number, the ring structure of the sterol. Okay, now in the previous membrane lipids, we have seen what are the individual components. We have, in case of glycerophospholipid, we have seen there are glycerol, then fatty acid, and then the polar head group. So what are the components of the sterol, or what is the source of sterol? Sterols are synthesized from a precursor molecule, which is a 5 carbon molecule called isoprene. Okay, so that is different from the rest of the membrane lipid we learned. Sterol is synthesized from 5 carbon precursor molecule called isoprene. Actually, this isoprene is synthesized from the very simple molecule acetyl CoA. So that we will learn during the synthesis of the lipid. Okay, so here we just need to remember sterols are synthesized from 5 carbon molecule isoprene. Not only sterols, other important molecules also synthesized from isoprene, and those are fat soluble vitamins, quinones, dolichols. Okay, now what are the functions of sterols, or here in case of vertebrate, especially human, we can say cholesterol. So cholesterol, we need cholesterol because many important biological functions are done by cholesterol, such as cholesterol is needed to synthesize steroid hormones and also to synthesize bile acids. So steroid hormones, they have important role in our physiology and reproduction, whereas bile acids plays important role in our fat digestion. Okay, so these are the sterols, but they are also membrane lipids. Okay, so up to now, actually the major two form of lipids we learned. The first one was the storage form, and the second one was the membrane lipid, that means the structural lipid. So lipids, we already at the very beginning we said that it is diverse in nature, so it has various functions. But only two functions or two forms of lipids we focused, but there are lipids which are like involved in other functions, such as functioning as signaling molecule, cofactors, and pigments. So these lipids which are functioning in these functions, they are required in very small quantities. That means if very small amount of these lipids are present, the function can be fulfilled. So we'll learn few of the examples. The first one, which are functioning as we can say the intracellular messenger molecule. So that is synthesized from phosphatidyl inositol biphosphate. So this is the glycerophospholipid, phosphatidyl inositol bisphosphate. And when it is hydrolyzed, that means it is degraded, we get two molecules. One is diacylglycerol, another one is inositol 1,4,5 bisphosphate. Okay, so inositol is kind of a sugar derivative. So diacylglycerol is released, which is having two fatty acids at the number 1 and 2 carbons, and in the number 3 carbon it has hydroxyl group, and the remaining part inositol, and then comes 1,4,5 triphosphate. That means 3 phosphate groups are attached to these carbons of the inositol. So these molecules then functioning as intracellular messengers. That means within the cell they are functioning as the signaling molecule. Okay, then comes other lipids, prostaglandins, thromboxanes, leukotrienes. In general, we can say the icocyanides, which are actually synthesized from arachidonic acid. And these are functioning as hormones, potent hormones. And then next, steroid hormones. We already learned that, which are synthesized from sterols, that means cholesterol, and they are also functioning as the biological signaling molecule. And best example of the steroid hormones are sex hormones. Then comes the vitamins, specifically the fat soluble vitamins, vitamin A, D, E, and K. They are also synthesized from the very simple precursor isoprene, like the cholesterol. And the vitamins, they have important function in our metabolism as well as physiology. And other molecules such as ubiquinones, plastoquinones, they are also synthesized from isoprenes, and they are functioning as the electron carriers, such as ubiquinones is present in mitochondria and functioning as the electron carrier. Sorry, plastoquinone, that is present in chloroplast, which is also functioning as electron carrier. So they are functioning as electron carrying molecules. And then some colorful or pigments, which are also lipids in nature, such as lipidic conjugated dienes. So they are functioning as the pigments, and they are present in flowers and fruits, or even bird feathers. So they are very colorful, and those are actually made up of pigments, and those pigments are lipid in nature. Is it clear?
Yes, ma'am.
Hello?
Yes, ma'am.
Okay. So next we will go So we will learn, actually not elaborately, very briefly, about the separation and purification of lipid, because we already come to know lipids are insoluble in water. So obviously we have to dissolve lipids in organic solvents when you are separating and purifying your lipids. And that depends on the polarity or solubility in non-polar solvent. That means whether your target lipid is soluble in the solvent or not. Okay, now the important thing that we already learned, that in the lipids there are different bonds are present, such as ester linkage, and in case of sphingolipid there is amide linkage. So we have to break down this bond, and we can do that by the use of acid or alkali. Excuse me. Excuse me for a moment. Okay. So like you have to break down those bonds, and you can do that by acid or alkali treatment, or you can use some enzymes, such as you can use phospholipases, because if there are phosphate group is present, then you can use phospholipases, and if there is glycosidic bonds are present, then you can use glycosidases. Okay, according to the what type of lipid you want to separate, you have to use that particular enzyme. Okay, now what are the name of the organic solvents we can use to separate and purify, especially for neutral lipids? We can use ethyl ether, chloroform, or benzene. Okay, now in case of membrane lipids, we can use ethanol or methanol. These two solvents are preferred in case of membrane lipids, because these solvent, they reduce the hydrophobic interactions, because the hydrophobic part of the lipid, they are interacting with each other by hydrophobic interaction, and sometimes there are also hydrogen bonds and electrostatic interactions. Those kind of bonds or interactions can be broken down, or it can be made weak by the solvent ethanol or methanol. Okay, as well as we sometimes the membrane lipids is interacting with the membrane proteins, so it will be easy to break down the interactions by using these solvents. Actually we have to use a mixture of different organic solvent, where we use chloroform, methanol, and water, and they must have this ratio. That means chloroform 1, methanol 2, and water is 0.8. In this proportion you have to make the solvent, and then you can use this solvent to extract your membrane lipid. So these are about the solvents and the particular reagent and enzyme you can use for the lipid molecule extraction or separation. Now the method which usually used to separate the lipids, that is actually called adsorption chromatography. So we already learned about chromatographic method. There we have learned different chromatographic method, and here this is another chromatographic method called adsorption chromatography. And we know that in chromatographic method you have a solid phase and a liquid phase, and you have to make a column of the solid phase. Here the column is actually made up of an insoluble polar material called silica gel, and this is actually coming from the silicic acid, and the structure of the silicic acid is here SiOH4, that you have to keep it in a glass column. That is your solid phase, and then you have to add your sample, that is your lipid sample, which are actually present in the solvent, which is here chloroform. So when you pour the lipid sample into the column, actually this silica gel is polar in nature. So if polar lipids are present, that means we can think about the membrane lipid, which has the polar head group. So they will interact with the silicic acid. But the other lipids, that means if the lipids are neutral lipids, they will not bind to the silicic acid, and they will actually pass through the column. So at the very first wash, we will get the lipid which are neutral, and in the column the polar lipids will be bound. So by this you can separate the two types of lipids from your sample. And the particular chromatographic method which is developed based on this absorption chromatography is called thin layer chromatography. Okay, thin layer chromatography is actually the application of adsorption chromatography. So this is one of the very simple and common chromatographic method for the separation of the lipids. Okay, next another chromatographic method which is also like I think very popular nowadays, that is called gas liquid chromatography. So here, so gas is present, that means your lipid or the lipid derivatives, they must have the feature, that means they must have the volatile nature. Okay, so if they have this volatile nature, then you can actually separate those volatile components, and again you have to pass through the mixture of volatile lipids through a column, and that column is made up of inert material, and in this case that inert material is actually an inert gas, for example helium. Okay, so your column is made up of helium, and you are passing your lipid volatile derivatives through the column. So the lipid which is more volatile in nature, that will come out through the column or will be volatile at the very beginning, and in this way they will be separated from each other, and that is known as gas liquid chromatography. Is it clear?
Yes, miss. Yes, ma'am.
So these are the two common chromatographic method used in separation of the lipids. The thin layer chromatography, which is actually based on the adsorption chromatography mechanism, and another one is gas liquid chromatography, which is actually done based on the volatile nature of the lipid molecules or lipid derivatives. Okay, now this is the last slide of our today's lecture.